Okay, um, it's 9 o'clock, so I'm gonna slowly start and we'll see if anyone else is going to join. So welcome to today's session. We're going to talk to the fantastic title, Future-Proof Your Genomics Journey. With me here today, I have two people from Penn Medicine. So my name, to start off with, is Fredrik Lysholm. I'm from Sectra, and I'm the general manager for our Genomics IT department. And we have, do you want to present yourself? No, okay. So, and with me I have Dr. Salvatore Priori, Associate Director in the Division for Precision and Computational Diagnostics, and David Lieberman, Associate Administrative Director from the same division. And today's session will cover first a little bit sort of introduction to what Sectra are and how we got into genomics, how we got started into this area together with Penn Medicine, and then sort of how we've transitioned and gone forward and what we are trying to do to sort of keep up with a fast-moving field. And hopefully we'll have some time for questions. There's a lot of things that we wanted to cover in this session, and we really struggled, I think, to leave stuff out. So, I mean, please do come see us at the booth if there's something that you want to ask additional questions for, or to see some of the ongoing development that we're not going to cover in this session. But first, a short presentation of Sectra. So I suppose you might not recognize Sectra. Sectra is a company that's from the beginning on the medical side, a PACS company. We started radiology 30 years ago, so we're one of the pioneers in radiology PACS business. Since then, we've grown. We were also one of the pioneers going into digital pathology. We've always strived to be a vendor-neutral IT company, so trying to provide a good IT system to allow our customers to basically choose whatever modalities and scanners, etc., that they want to use. We've been sort of fairly heavily promoted by the Class Institute that ranks vendors in the PACS business. I'd say basically just for following our core values, which is about trying to listen to our customers, don't overpromise, and try to deliver of what we say that we're gonna do. And we're one of the most awarded in this area in the PACS business, but fairly new in this area. The company has basically two areas. So we do work with secure IT and IT communication systems, and we do work in medical imaging IT. And then we have the new area, which is Genomics IT, that we've started to sort of broaden our portfolio in the diagnostic space. And to sort of set it a little bit together of what we're doing. So I said before, we're trying to work with our customers and really follow and predict trends. And that's sort of taken us from radiology into digital pathology, and nowadays more across to think about diagnostics from a broad standpoint. Obviously, we're trying to work with cross-discipline workflows and really how to utilize the patterns and this sort of idea that we have a lot of diagnostic information produced in different diagnostic disciplines and how we can use them together in our system. And in the end, it's about reaching the optimal treatment decision for the patient, right? And if you want to see how we're working a little bit with cross-discipline workflows, that's also something that we're not going to show today, but please do come by the booth. I want to say that sequencing diagnostics sort of in molecular pathology is still kind of a fairly new thing. I mean, and we've seen the growth of a new diagnostic area multiple times. It's the same time like when the radiologist department gone from analog to digital back 35 years ago, and we saw the same sort of trajectory with digital pathology, and now it's the same thing in molecular pathology again. I mean, we started something new, we had new requirements, and we tried to cope with it in the best way we could, which meant, and still means, that we had to build a lot of stuff on our own, trying to get stuff to work together. And eventually, it's growing, it's getting bigger, and it becomes a problem. And I think that's sort of where we saw the market were. We went around talking to a lot of different customers that sort of had the same problem. What about molecular pathology? How do we handle that? How do we handle the transition from digital pathology into molecular? How do we do this in an efficient manner? We have all these scripts, and I've talked to multiple CMIOs that said pretty much the same thing. This part of my IT organization is really the wild, wild west of diagnostics. We have a lot of things, and if that guy would leave us, then we have an issue, because he's the only one that understands how this system is going to fit together. So I think this is where we can help, what we've done before and what we're trying to do. And we sought different customers to help us on this journey. And fortunately, we found Penn and started to work on a collaboration project, basically to see, okay, how can we help out with IT software in this space? And I'm not going to talk anymore and leave over to Dr. Priori to sort of tell you a little bit more about that project and what we've done. And yeah, you can use that one as well if you want to.
All right. Good morning, everyone. I'm going to give just a brief overview of our collaboration with Sectra, and then we'll launch into sort of, giving you a high-level overview of the product that we developed together. But, as Frederick said, this space is relatively new in medicine, and Penn Medicine was trying to be at the forefront of this and develop a genomics laboratory, mostly for cancer oncology, and trying to figure out how to manage all of the requirements, all the data, all the workflows that go into running a complex operation like that. And so, we think about clinical genomics lab in terms of the phases of testing. So we think about pre-analytical phase, which includes sample collection, transport, nucleic acid extraction. Think about the analytical phase, which includes the library preparation and the actual sequencing, and then the post-analytical phase, which includes the data analysis, results interpretation, and results reporting. So all of this together has huge data and information requirements. We have to receive orders, we have to accession samples, track them through the lab. We have complex laboratory workflows. We have to receive sequencing data from the instruments. We have to analyze that data, annotate it, and curate the variants that come out of it. And then we have to generate the report, and then transmit it to the electronic medical record. So before we were working with Sectra, a lot of genomic laboratories, especially in academic settings, were kind of on their own to figure out how to manage these workflows efficiently. And so those of you familiar with this space, this may not be such an uncommon arrangement to have a completely independent database that manages your genomics data. For us, that was a product called FileMaker Pro, which is just a general database software, not specifically made for genomics. And we configured it to meet our needs, but really wasn't ideal. As Frederick said, David and I were the ones managing it, and so if anything happened to us, nobody knew how to do anything with it. And that didn't interact with our laboratory information system, which is Cerner, and we had to manually sort of take things from FileMaker Pro, put them in Cerner, and Cerner has an HL7 interface with our electronic medical record, which is Epic. So really not an ideal situation from an integration perspective and from a maintenance perspective, because we were doing that, and you were relying on a couple people in the laboratory to keep that going. So really, the point I want to emphasize here is that when we joined with Sectra, the development was sort of from scratch, and it really was based on the clinical needs. And so we articulated to Frederick and his team that we needed an end-to-end solution for clinical genomics testing. And so that's what we have now. It launched in sort of mid-2024, and we continue to use it now. But all of this stuff, we get our orders from our EMR now. It flows into the system, and we'll kind of show you the different modules that track every single step in the clinical genomics workflow. It's completely integrated with our instruments and receiving sequencing data from our bioinformatics pipeline. And then at the end, it's integrated into the EMR to deposit PDF reports and discrete genomics data. So we have a lot to show today. I can talk a lot more about the collaboration and how we developed it. Happy to do that after the session, but I'd like to sort of jump in and give you previews of the features of the product we developed, and then we'll sort of segue into things that we're developing now after the product launch. So with that, I will hand it over to my colleague David, who will talk about some of the wet lab workflows that Sectra manages.
All right, thank you. Couple things to note. I did already ask them to install people movers, so hopefully they'll get those installed. Not funny. The other thing I want to note is that there is some fake patient data that you'll see in some of these. It's fake, it's not real PHI, so just keeping that in mind as well. So I want to start with some of the functionalities that we see in a browser-based system that are extremely useful when moving through the system. So it's browser-based, so we can use our forward and back navigation buttons to go, whoop. You like that, right? Our forward and back navigation buttons to go the right way we want to go. We can switch it between light and dark mode depending on our visual preferences. We can share URLs directly to a lab batch that we want. So I want Sal to look at this extraction batch. I just pop the URL in our chat, and it opens the window right up for him. So it's really convenient. It's browser agnostic, so it doesn't matter what you like. You can use it, and the system will work just fine. And then having the ability to have multiple tabs open so I can compare two runs that I want to compare side by side, things like that are really useful. I wanted to move into the wet lab, both the order management and the wet lab management next. Really high level slide here where we're seeing a lot of the steps that we see across the order management and across the wet lab management. So in order management, high level, we're handling incoming clinical orders, and we're able to queue validation samples in this same section. So we're tracking pending samples that come in, like Sal said, directly integrated from the EMR. So those come in with all the patient sample order information directly. We can see them waiting in our queue, and so we know that they're coming to us even if we don't have them in our hands yet. We can real-time audit the information on these order screens. So as we get a lot of the information directly, but we do have to add in some additional information, and any changes that are made are audited, and we can see that in the screen, what's removed, what's added. We have automated workflow assignments, so we set up our workflows, we configure it. So based on the test order, based on the sample type, it is automatically going to flow into the appropriate queue in the lab, electronic queue in the lab. And like I mentioned, we can also set up workflows for validation samples. So we need to validate reagents and put in samples to run those validations, to validate new assays. We can set these samples up so that they go through the process to the appropriate level and then get kicked out of the workflow automatically. So I might only want to have some samples to validate my extraction workflow, and those will get kicked out of the workflow after the extraction step, and then I can analyze the data. Looking at the wet lab management a little more specifically, we have our lab workflows, our reagents, our instruments, our indexes, all need to be tracked well here. So we have our reagent management. We're tracking amounts, the validation status. Has this reagent been validated yet or not? Is it expired? And then assay association. We can associate specific reagents with specific assays. We can configure the workflows. I said this a number of times already. But we are building these workflows so that they meet the needs of our lab. And as we develop, and we just have recently developed some new assay workflows, we can integrate those into the system as well. There's system-assisted workflow regulation. So we can configure hard stops. We can configure warnings. So if we need our techs to input this data, it forces them to do it. Or it might be this is strongly suggested, and it's just maybe a warning in this situation, and in some circumstances it's appropriate, and some it's not. And then they're required to sign off on each batch that they, throughout the whole process. So that's at the wet lab and then in the dry lab steps as well. So you have to input your credentials to sign off, and so we can audit that way as well. We have a queue that we manage samples that need some additional human input to determine how to resolve them, and so we call that the issue resolution queue, but it's an additional queue. If a sample fails, for instance, maybe it just gets re-queued right back to that work queue, but it might require some additional input by the lab supervisor or somebody, and so it can populate this queue, and we can see if we need more material or if we need to put it onto a different assay, something like that. I mentioned some of the quality management pieces here, but another one here is it forces unique index assignment, things like that that are really important for an NGS lab. So looking at as we drill down into the screens a little bit more, so what you're seeing here on the left-hand side, so these are a number of queues that we have when orders come into the lab. So the first one that's highlighted there that we're looking at is our new unverified. So these are electronic orders that have come in. We know nothing about them. We don't have the sample in our hand. We just have the order placed. So we know it's coming. You can see on the left-hand side as you progress further down, we have samples that says the next queue is awaiting samples. So maybe we reviewed the order. We know that we've input additional data that we need. We've approved this order and said, yeah, this is good to go. Or maybe we need to do some additional follow-up with the clinician before we approve this order. But we can do that before we even have the sample in our hands if we want to. Then we can once we have our samples, it can be released into the lab and progresses into the in progress. And you can see we have some additional queues like our hold queue, validation review queue. When we look at the center section here, this is where we have this is the actual queue of samples. And if you can see patient information, you can see order identifiers, things like that. And then when you click on a specific sample, you can get some more detailed information here in the right pane. What's nice is that all of these queues are filterable and sortable, and Sectra gives you some pop-ups here too to help you understand better ways to filter and sort. So if I want to do a not search or not filter, I can put in an exclamation point and do that type of filter. Now I've drilled down into a specific sample. On the left-hand, same overall format. On the left-hand side here, you can see patient historical samples. So I'm on the green sample, and that's the case that I'm looking at right now. But I can see all the testing we've done for this patient over time in the past, and some, I'm looking at a sample from yesterday, and maybe we got a couple more samples today. And you can see that here. And I can easily navigate, click on these and navigate across. We can also insert patient events here as well. So for instance, and I think Sal's going to show you this more too, but for instance, if they had a bone marrow transplant, we can insert that here and we can see where that occurs in time as well with our testing. All this electronic information is coming over automatically through HL7 with the order. So the patient information, the test order, the sample information, and then over here on the right, you can see the auditing of the events that have the data that has changed manually in this case. Okay, so once the person receiving the sample releases it electronically into the lab and sends it back there, it's now going to land in that queue accordingly. And so you can imagine, or you don't have to imagine, probably many of you know, there are simple lab workflows, there are more complex lab workflows. And so this system really allows us to manage both of those scenarios very well. So many of our workflows are more complex, and so we start on the bottom here with a single sample, breaks out into multiple extraction events. Those individual extraction events can break out into multiple assays, and the system can allow us to manage that easily and automatically. So on the left-hand panel, that's an example of how we have some of our workflows broken out. So you have your different extraction events that you may carry out. From there, once they're released from that, they'll feed into various assay workflows, and then finally coalesce back into a sequencing step. When I drill down into one of these steps, that's what we're looking at here, and so you can see the center section again is our queue of pending samples. So these have not been brought into this extraction batch yet. They're ready to go though. Once I'm ready to start a new extraction batch, I can bring them over here to start it. I can do it a number of ways. I can drag and drop individually. I can select a set and drag them all over. I can just say fill queue, and we have pre-configured a maximum amount that we can queue up and just press a button and it'll fill with the first set that reaches that volume of samples. And then I can drill down into ongoing events as well. So these are already in process. So if we drill down there and we see a library prep that's ongoing right now. On the left is the input samples. On the right is how our technologist has plated them in the plate, the visual plate here. And as they do that, it provides some text on the bottom there of this sample was plated into this well, and you can see that. And so as I move through with my cursor on each of these, it identifies which well position as well that the sample is in. I'm adding controls here that I've input into the system prior. We have a set of controls, and so the technologists can add their controls, can add a well with an NTC in it, and a template control as well. Here we're tracking our reagents, so this is critical for any lab. We need to know what lot of reagents we're using, what their expiration is. It's going to tell us the expiration date here as well and flag something if it's expired, if it's not been validated, all of that gets flagged. There's also instrumentation here that we're tracking. Skipped over, but there's a master mix tab here as well, so that will automatically calculate the reagent inputs required based on the batch size and based on a certain pre-configured overage that we want to have as well. Here we're now on identifying sample data in QC. Each line is a sample, and we have boxed here the input initially. So this is the input volume of sample, the input water, so that's all tracked here in the system. And then we have the output. So we have certain metrics that we're outputting. We have volume that we're outputting from this step. We have our index tracking. So we can, based on the lab step, it knows which index sets we need to have for this lab step. We can pick our starting index and then just automatically fill from there. We have the ability, though, to pick and choose as well, one-off if that's what's wanted. And then the last piece on this step is it's really important to communicate across the lab, so we have opportunities for overall batch comments, we have opportunities for individual sample comments. All of these are going to be visible to the person reviewing the case at the end as well, because they need to know, was there something abnormal about the extraction event that happened, about the library prep, so that's really important. Once we complete our sequencing, once the sample goes on the sequencer, the next step is going to be tracking bioinformatics. And so this, like Dr. Priori said, the Sectra Genomics is integrated with our bioinformatics pipeline, and so it knows that there's an ongoing bioinformatics pipeline event for this sample. Once that's complete, it gets brought in, and these samples are now ready for our QC management now that the data has been brought in. And so that's what we're going to look at here. So we now have a sequencing run that has come into the system. On the right-hand side here, we have the metrics that come off of the pipeline, so our total reads, our mean coverage, those types of things. We have a sample here that unfortunately failed below our mean coverage. So we have configured this so that when it falls below a certain level, it is flagged red. So you can see that red value there, so that stands out. The system is also on the left here suggesting that this is a failed sample. It automatically does that based on our configuration. So it still requires a human to go in there and say, yes, this is failed. I agree with that. It's not going to automatically do that, but it's telling us this sample is suggested to have failed. This is why we're suggesting that in the system, and then we can go from there. And that's what we're doing here. We're marking it as pass or suboptimal or fail. We're doing some additional quality checks on SRY, and so if this is a male, do we see that in the sequencing files? If it's a female, do we see that represented in the sequencing files, additional QC? We're also tracking our reagents, so did we use an unvalidated reagent in this lab step? And so the system will show that here, that these reagents were not validated at the time these samples were put on. And it forces us, we've configured it, it forces us to mark that these reagents have now been validated before it allows us to sign out in the case. Okay, I think this is the last thing I'm going to talk about before I hand it back, but this is a really important piece. So this is a tool that is built in to allow us to look for potential contamination in the system. So what we're looking at here is sequencing run, and any variant that was called by more than one sample or that's present in more than one sample on the sequencing run. On the left are the variants, on the right the columns are different samples and the allele frequency of that called variant. So we can do this by sequencing run. We can also choose to do this by other lab steps, by extraction, by library prep, and sort by that. And if we suspect a contamination event that happened in extraction, we can look across all the samples that were extracted together, even if they're on different sequencing runs. So here we have one where I'm a little concerned. I'm seeing this EGFR exon 19 deletion in 2 different samples, both at the same allele frequency. So I'm going to dig into this a little bit more, and I'm going to review the benign SNP profile. And I see that they're essentially identical for these 2 samples. So that is a huge red flag. Dig into this deeper, and we find that there was, in fact, a lab error where the technologist input their sample into one well, and then instead of the next sample, they re-input that sample into the next well. So immediately upon looking at this data, it was clear that there was an event, and we had to follow up. So really, really useful to identify those. Here's another example. We started to review. We see a number of tier 1, tier 2, tier 3 variants shared by 2 adjacent samples. Again, super high level for concern right away. That's obvious. Dig into the SNP profile, and they're different. So we did some additional due diligence, and in fact, this is just a rare circumstance where we had common variants, but the SNP profile told us that these don't appear to be the same sample. We did additional due diligence, retesting, and it held true. Okay, so that's my piece for now. I'm going to pass it back to Dr. Priori to talk a little bit more.
So yes, there's a lot that goes on before cases reach me for interpretation and reporting, and I just have to emphasize, the complexity of that workflow and having everything managed in a consistent and robust way in the system certainly gives us a lot of confidence at the end of the process that things are safe and efficient. And all the regulatory requirements that go into managing a genomics laboratory, it makes implementing those requirements and tracking those requirements much easier, which is great. So I'm going to talk a little bit about the diagnostic reading process. So once the wet lab process is done and the sequencing data has been received, David was talking a little bit about how we review the quality of that. So our process in our laboratory is that we have variant reviewers that review the quality metrics for the run. They'll annotate the variants, they'll curate the variants, and then they'll draft a report for us. And then the reports come to me or one of my colleagues, and we review and approve those draft assessments. We'll add additional interpretive text and comments and summaries, and we'll complete the report. And then once we verify it, it'll go into the EMR. And in unusual circumstances, sometimes we need to amend reports, and there's a little bit of back and forth in this process. So I'm going to just demonstrate some of the key features of the product that help us during this interpretation and reporting part. So there's a lot on the screen. You're not meant to see everything in detail, but I'll walk through the fact that everything you need to do this process is on this screen, which is very nice. You don't have to navigate between different views. So in the middle, we have our list of variants that are imported from our bioinformatics pipeline, and I'll talk a little bit about the organization of them. And then on the right, when we have selected a specific variant, you have detailed information about that variant and its curation and annotation on the right-hand side. So on the left-hand side of the screen, there's detailed information about the sample and the patient. So as David showed, on almost all of our screens, we have a history for prior testing for the patient or concurrent testing for the patient. And we have the ability to enter important clinical events for the patient. So David alluded to the fact that you can enter in a bone marrow transplant for a patient with a hematologic malignancy. And that makes a big difference because from that point forward, you expect a mixture of variants from donor and recipient, and potentially, the goal is that they'll be all recipient at some point. But without knowing that information, you might be worried that something's amiss with your specimen because you have variants at allele frequencies that you wouldn't expect in a sample that's derived from one person. So here's just showing you the bone marrow transplant entered into the patient history, and then we have that moving forward. Okay. So on this screen, we can see, we're looking at a specific variant, and one of the things that we want to do is look at the actual sequencing data associated with that variant. So Sectra is synced with Integrated Genomics Viewer, or IGV, and so you can actually just pull up your sequencing data, and as you scroll through your list of variants, it will sync to the appropriate genomic position, so you can see the sequencing data and evaluate it to make sure that it's appropriate. And so you can see that as I move through the list, IGV will sync automatically to that position, which is very efficient. On the right-hand side, we have the detailed information about the variant, as I mentioned. And a lot of it is collapsed, so you have to expand things, but you can see detailed information about what the bioinformatics pipeline sent over about the variant. And then we have very helpful link outs if you wanted to go to NCBI to look at details about the gene or details about the reference transcript that it was called from. And then this we use all the time. It gives you a inventory of every time the variant was called in your database. So you can see which cases had this variant and what allele frequencies they were called at, and we have some summary statistics up at the top, the mean depth, mean VAF for those variants. And you can see how they were classified as well. So if classifications change over time, you can see that progression. And while we have a bioinformatics pipeline that annotates our variants for us, you have complete control to change the annotation of your variants. So you can change the C dot, the P dot, you can change the reference transcript that you want to annotate from. And it also supports alternate nomenclature, which is important for legacy transcripts or legacy nomenclature for specific variants. One of the main tasks that we need to do as we're curating the variant and deciding what its significance is and its classification is reviewing external databases. And so that can be pretty time consuming if you don't have a structured way to query those. So the system is configurable and allows you to set that up. And then you just click the boxes that you want. You hit this, I don't know if I have you hit this little button here and it'll just pull up all of the sources that you want. And on the back end, you can configure whatever you want. So it's a structured URL query, and as long as the outside source will accept it, you can use these nice, handy little smart phrases that are constructed to query chromosome position, genomic position, C dot, P dot, whatever you want. And so it's a really handy way to semi-automate a lot of this repetitive work that we have to do with variant curation. All right. And just to note, we have some tiers here that you're seeing on the screen, but the system can support any classification scheme you want. It's fully configurable. So if you want to use the AMP criteria, you can certainly use them. If you don't want to use them, you can use something else. It can support pathogenic, likely pathogenic, VOS. It can support this. It can support your new classification system that you invent and promote to the community. So again, just along this theme that it really is customizable to your lab's needs and your clinical, your local clinical situation. And then one of the other nice features about the system is the ability to group common variants together. So variants that share common interpretation and common clinical significance, regardless of their individual C dot or P dot, can be grouped into what we call a categorical interpretation. And you can configure how it appears on the report, and you can tie a specific category or classification to that whole group. So when you add the variant to that group, you don't have to do any further curation to it. It'll just automatically inherit the curation from the group. So the example I have here is exon 19 deletions, which is a recurrent change in non-small cell lung cancer and EGFR. And it's really handy because there's a lot of different variants that all fall into this category. And they have the same sort of clinical implications that we want to convey to the end user. And so you can see here, this is how it would appear in our reports. It specifically says in the report that it's an exon 19 deletion, and then you can structure the interpretation to say, hey, there's FDA-approved drugs that are approved for patients that have this specific change. One of the things that we do, especially in hematologic malignancies, is we have repetitive sequencing for the patients, so they don't get just one study, they get multiple studies over time. So we have a grouping in the review section of previously reported variants, which is very helpful. So those will just group at the top, and so you can immediately see whether you're seeing the same variants that you saw in the past. If you're missing any, or if you're seeing new variants, you'll see them sort of outside of that group, and it's very easy to appreciate when things are changing for your patient over time. And then we have another configurable section of the report that we call review lists, and review lists are very helpful because if we want to manually review specific areas based on a diagnosis to make sure that we're not missing anything, we can do that. And so this is just an example of our review list for AML. It has us reviewing very important biomarker areas for that disease. Okay. And then we have a ability to filter down this list so that you can be more efficient with your review. So if you want to filter out your tier 4 variants because you don't want to look at all of those, you can just hit this button and you can condense your review list down. And then another handy feature for looking at variants longitudinally over time, you can pull up this view where it shows every variant that's in the profile, and it'll show you if you had it in a previous sequencing study, and if you did, it would have the VAF of the variant at that time. If it's missing, you'll see a blank space. Okay, so there's a video in here somewhere, so let's see. We also have functionality for kind of more complex variants besides sort of single nucleotide variants and insertions and deletions. So this is just showing you the functionality that was built out for copy number variations in Sectra. So you can see that we have this copy number variant viewer that shows you the copy number variant data. And I think this video is just kind of going through and showing you what you can do in this view. So you can zoom in, you can zoom out. Genes are annotated at the bottom, so you can see where your copy number data overlaps with specific genes. And you can go up to the top, you can zoom out, and you can see the whole genome view. You can see your individual probes that are providing the data for your copy number calls. And I don't know if it already showed it and I missed it, but you can search at the top based on gene or genomic position to get to a specific area of the genome. So very handy way and efficient way to look at complex genomic variants. And then finally, once you've reviewed everything, annotated it, you get to your review screen, and there's a lot of abilities here to add interpretive comments, summaries, and nicely, the system provides configurable soft and hard stops. So for instance, Dave was talking about the QC part of the workflow. If somebody didn't sign off on that batch, this would throw a hard stop and I wouldn't be able to verify the report until somebody goes back and verifies that the quality control was okay for that batch. So it really allows us to prevent mistakes in terms of that, or omissions of parts of the workflow that are essential before we report things. And then you get a nicely formatted PDF. This is completely configurable by implementing a Word template into the system. And then when you verify it, you can either just close out the case, or you can send it back to an issue resolution queue if you want to reflex the specimen for further testing. So there's a lot of ability to direct samples to previous steps in the workflow if you need to do that. And then once it goes over to the EMR, you get the PDF, but then you also get discrete data deposited. So, sorry, Frederick, I didn't mean to hit you with the laser beam. The EGFR example I gave, for example, this is how it would populate in our EMR. And that's nice because you can start to do things with discrete data like this, like configuring clinical decision support in the EMR, where if the exon 19 deletion tag is in there, you can trigger a alert to the ordering provider that there's FDA-approved therapies associated with that biomarker. Okay, so that's like a high-level overview of the core features of the product and how it helps us manage our day-to-day workflows. Since we've launched it, we've continued our collaboration, and as clinical need has arisen, we've continued to develop new functionality, so we're going to talk a little bit about that. But definitely go down to the booth in the expo hall because they'll have the computer set up and you can actually interact with the product and see these features for yourself. And that's a better way than kind of going through the slideshow. So yeah, definitely take advantage of that opportunity. So I'm going to pass it off to David one more time. He's going to talk about some of the things we're developing for the wet lab side. And
Try to move through a little rapidly. I know we're moving towards the end of our time, but wanted to highlight a few safety and quality pieces that the system also has within it. So volume, TAT, we have a dashboard for tracking all of these required metrics that we need to keep track of for both for regulatory reasons, but also to make sure that we're managing our systems well and our turnaround time well. So there's a lot of metrics here, and you can see I can drill down into this, and I can look at my ongoing volume by test. I can look over specific time frames. I can export the data. I can copy the graphs here if I need to present it to my lab director, to whomever, my quality manager. The system uses institutional credentials with role-based permissions, so our access to Sectra Genomics is integrated with our institutional credentialing system. So I don't have a separate password here. And when my password changes for Penn, it changes for the system as well. Also, as it's your access, your read and write access is all role dependent. So as a technologist, I can't sign out a report. The system won't let me. As a faculty attending, I can't create a lab batch. The system won't let me do that either. And these are configurable as well. There's work queues present, so as a variant analyst, I can see what cases are assigned to me quickly and easily as soon as I sign in. There's communication within the system where I can send, as an administrator, can send a message to my team that's important. So in this case, we have we have some updates to one of our SOPs. I need our technologists to sign off on that before they initiate their next assay. So this is how I'm setting it up. I'm setting it up to expire on a certain time. I'm setting it only to go to my technologists. And then this is what they'll see. When they log in, they have a big blue banner that says, you need to do this. And I can send it to just technologists. I can send it to everybody. I can send it to technologists and variant reviewers. So it's configurable in that way. We've used this for some of this presentation, but if I want to present, if I have a vendor that I need to look at some of the data in the system, I can click a button. This is a video too. You can see my patient data. I click this button, and it just hides the patient data everywhere in the system. And so that makes it helpful for presentations and for showing the team. All right, that was a couple things. Pass it back to Dr. Priori. We're going to keep running back and forth.
Yeah, so some of the things that we've been developing to improve our reporting. One of them is cancer risk variants. So we do somatic testing in our laboratory, but there's recommendations now that you're going to pick up germline variants that predispose people to hereditary cancer syndromes, and that they need to know that so they can be referred to the proper genetic counseling resources. So that has been a manual process between us and our medical genetics department for a long time, but we've developed stuff in Sectra now to make that a little bit easier. So On the back end of the product, you can actually say, okay, well, BRCA1 is a variant that if we detect that in a sequencing panel, we want to make sure the patient gets genetic counseling. And so you can actually structure a description for BRCA1. You can flag it as a cancer risk gene, and then you can configure the categories. So you can configure conditions for a variant in that gene that it'll flag it as being a cancer risk variant in the report. So here we're just saying, if the category for BRCA1 is tier 1, I want you to put in a cancer risk flag for this patient. And so in the reporting, what you get is this cancer risk variant thing that will, text that will give you a description of that. And in our case, our implementation is please put in this order into the EMR to get medical genetics referral. One sec, I'll finish up and then happy to take questions. Related to the donor-recipient issue that we talked about before, post-bone marrow transplant, germline variants that are coming from the bone marrow donor have different implications than germline variants from the recipient. So actually, you can put into the system, after a bone marrow transplant event has been logged for the patient, you can tag whether a variant is coming from the donor or the recipient, or you don't know. If it's coming from the donor, then a different text will flag into the report, and it'll specify that it might be donor-derived and has different implications than a recipient-derived variant. We spent a lot of time developing hierarchical diagnoses. So this is our implementation in the system at the moment. It's based on OncoTree, but you can modify it however you want. You can start from scratch and make your own. It's just showing you here some of the things that were modified, things that I added that are completely new, that are local, things that don't have a flag next to it were imported with the tree. And the nice thing about this is that we can tie a lot of the functionality in the system to the diagnosis tree. So here, I'm just showing you an example of how we can change the tiering based on the inputted diagnoses. So here we have a tier 1 variant for this KRAS G12C because it has a non-small cell lung cancer diagnoses. But then, when you look at the configuration for the variant, you can actually tie it to specific diseases. So non-small cell lung cancer and everything nested under there will get a tier 1 category for that specific variant. And you can add other diseases. So we have I can't read it completely. Colorectal adenocarcinoma is also tier 1. And then anything else that is in the system when that variant pops up would get tier 2. So there's sort of a general category, and then there's more specific categories, and you can configure that however you want and is appropriate for the specific variant. Okay. So just a couple of the things that we're developing to keep up with a moving field, and I'll just hand it over to Frederick to wrap up.
Okay, so we're almost running out of time. We really struggle to see what to cover, etc. I'm just gonna say, from a vendor perspective, we really had long thoughts about how do we do to actually support a moving field, how we do it operationally, how do we do it from a technology standpoint. And what we're really trying to do is build for the future and build for having to support a fast-moving field, which means that we're providing this as a service in terms of how we deliver the product to our customer. And you can see since go live with Penn, we've rolled out more than 30 releases. So we're sort of trying to continuously develop and do new software for our customers. And sort of our main idea here is really that we're trying to develop while new features are needed. So that's really what we're trying to do. And we're a vendor-neutral IT system. We want to help our customers basically use whatever you want to use, what's the new modality instrument at the time. And Penn are using different modalities from different vendors. From our Sectra perspective, that's really not our thing to say. We really want to facilitate whatever you want to and make sure that we have the right sort of operational and technical abilities to make sure that we can deliver this on time for you as fast as possible. So our vision, like going forward, is really to try to capture all the genomics data in one system. And as I said, develop on new assays and diagnostic reporting features as we go. This today, and I mentioned it in the beginning, we're a PACS company from the beginning. This today is part of our Sectra One cloud offer, and among that is also radiology and digital pathology. And we've sort of just started off in this new space and are trying to pick up as fast as we can and add on new features, but also to try to work on additional fields, sort of additional cross-platform workflows. And again, I mean, we set out from the beginning, as Dr. Priori said, to really build something that's for clinical diagnostics first. That's where we want to support, rather to have something that was initially built for academia and try to retrofit it for a clinical setting, which means that we put a lot of emphasis in safe, efficient workflows within the lab to serve the patient. And that's, thank you for today. I think just on the second, perhaps 1 minute over. And we don't have a lot of time for questions according to the time, but if you want to, please do ask questions and we'll stay. And if not, come see us at the booth. Thank you.
So, no, but that is certainly possible. And the reason why the answer is no is just because we have some upstream workflows that were already present in our order processing that sort of catches that stuff ahead of when it comes into Sectra. But if that was not something that we had already had sort of people managing some of these orders upstream of us, would have been a need that we would want to catch and configure into the system. So that's absolutely within bounds. It just wasn't one of our pressing clinical needs, so we didn't develop it. But yeah, that's very doable in the system.
It's easy to see, you can see in time where things have come in, and so I can see I just got an order a week ago and it's still in process. So, but yeah, building in a flag that automatically stops it could be helpful.
Related to that question, you started to talk about that you mentioned that you're getting information from the EMR and from Cerner. How much of that is bidirectional? And the reason I'm asking, connected to the last question, is that you talked about having all the other data from your lab, all the NGS data. But as we all know, it might be relevant in the interpretation, other molecular tests that have been performed, cytogenetic tests, IHC. And so how much can you extract right from the EMR versus needing to look up in the EMR, quote unquote, manual?
Yeah, it's a good question, something we've been thinking about. We've had a lot of sort of bigger ticket items that we wanted to develop in terms of just routine functionality, but we've been having those discussions actually because there is a lot of data that could be coming from the EMR directly, so it doesn't have to be manually input. So, other tests that are associated with that encounter date, are easy to pull in, and so all of that is doable and is on our sort of punch list to implement, because that would save, time for somebody manually inputting them, and then cuts down on errors in terms of, transcribing that stuff manually from one system to the other, so.
It's one of those items we've talked about, like possibly investing time to go into a little bit more once you come over to Epic fully. So we do have the ability to fetch some information from Epic in that regard. So it's definitely something that we should investigate further.
And then relatedly, if you catch an accessioning error, again, how much information can flow back into Epic, say, if it was a mistake in Epic that needs to be corrected, can any of that be automated, or is that something that you still need to separately?
At the moment, that would be separate. So like, it couldn't go in and change the encounter at the moment, or the order, the specifics of the order to correct errors that were input at that stage, so.
So we have examples of, we've done some testing, there was subsequently an error identified in patient data in Epic. It was fixed. Next time we got the order, it updates the Sectra system with the correct information. So that's not quite the same thing, but yeah.
Yeah, but we actually caught a date of birth error in Epic, which I had never seen before, and so we were able to correct that by, separately in Epic, and then the correction flowed through into our system. Okay. Any other questions? Did you have a question? Oh. We use our own, we use a variety of pipelines depending on the assay that we're doing, but Sectra doesn't run our pipeline for us, it just receives data from it, if that was your question. Yeah. Yep. Did you have a question? So, yes, that's very, very observant. I'll switch that out to a more convincing BRCA variant in the future. We have a sort of a VAF agnostic referral policy at our institution. Part of it is because, you can have copy number variants that deviate the variance from germline expected values. But that was like a lot of discussion between us and medical genetics, and they're like, no, we just want to know everything. So if you see one that falls into this category for any of these genes, we want a referral, and then we'll decide whether the patient needs counseling.
The risk of unnecessary patient harm, like just from the anxiety associated with this, like I would urge you guys to try to look at it from make it more of a database, like looking at the evidence to support something like that, where you're going to have a lot of false positives
Mm-hmm.
and the potential risk associated with that.
Yep.
And then I guess I was also curious, like if you guys, because on that one slide, showed the specific BRCA1 variant, you just, because there could be lots of different, there's like infinite possible number of potential variants.
Yeah, right.
And then how that's going to be interpreted. Because there may be like, kind of well-known, mutations that are known to be germline pathogenic, in which case, yeah, maybe even a lower native makes sense.
Yeah. I mean, we're sort of deferring to our medical genetics colleagues and also some of the guidelines that have been put out in terms of recommendations for referring patients who have specific types of variants that are discovered on their somatic panel testing. But agree with all of your points. Those are concerns, right? It's going to potentially alarm patient that they might have something that they don't, and refer people to maybe some unnecessary testing. But yes, but you can configure the system to do whatever you want. So this is just our local implementation, and I'm not saying it's perfect or ideal, just what was developed in collaboration with them. So, but not a necessary feature of the system. All right.
Cool.
Thank you.
Thank you.